Hi everyone! Today we're going to talk about enzymes. Enzymes are a kind of protein that act as a biological catalyst, which accelerate chemical reactions. Enzymes are important to humans because they do the job of building muscle, destroying toxins, and so on. One of its functions is to break down food particles during digestion. Amylase, maltase, lactase, lipase, and proteases are some types of significant digestive enzymes. Zymase is the first discovered enzyme, which naturally occurs in the yeast and helps in fermenting the sugar molecules to produce carbon dioxide and ethanol. In 1897, a German chemist named Edward Buchner converted sugar molecules into ethanol with the help of zymase derived from the yeast, and this became the foundation stone for modern in vitro enzymology.
An enzyme is a macromolecule that acts as a catalyst, a chemical agent that speeds up a reaction without being consumed by the reaction. Enzymes could modify specific substances called substrates. They are very selective in who they bind to and modify. This is the enzyme's specificity. Here comes the question: how does a tiny enzyme speed up the reaction? The answer to this is simple. Imagine you are making a snowball in your backyard, and your backyard is a giant hill. To make the snowball, you will need to push it all the way up over the hill first. That is not an easy task, because you will need to push very hard to make it happen. The initial requirement of energy for starting a reaction is called the activation energy, labeled as EA. The energy must overcome this EA so that the downhill part of the reaction can begin. But if the hill in the backyard is slower, you will not need that much energy to push it upward. An enzyme is a very helpful body. It catalyzes the reaction by lowering the EA barrier so the reaction does not need to absorb as much energy to reach the summit. It is also crucial to note that an enzyme cannot change the delta G for a reaction, which determines the reaction is whether endothermic or exothermic.
Enzymes are folded into complex three-dimensional shapes that allow smaller molecules to fit into them. The place where these molecules fit is called the active site. In the lock and key hypothesis, the shape of active site matches the shape of its substrate molecules. This makes enzymes highly specific. Each type of enzyme can usually catalyze only one type of reaction. Some may catalyze a few types of reactions. The diagram shows how this works. In the example, the enzyme slipped one molecule into two smaller ones.
Enzyme activities can be affected by a variety of factors, including temperature, pH, and concentration of enzymes or substrates. Enzymes work best within their optimum temperature and pH. Otherwise, the ability of enzymes to bind to a substrate will diminish. First, let's talk about how temperature affects enzyme activities. In most cases, rising temperature speeds up the reaction and lowering the temperature slows it down. However, if the temperature gets too high, the enzyme will lose its shape and stop working. It is known as denaturation. Each enzyme has its optimum pH range. For pepsin, which breaks down protein, the optimum pH is 2.5, whereas trypsin, which serves the same function, has an optimum pH of 9. Changing the pH outside of the range will slow down enzyme activity and even denature the enzyme if the pH value is too extreme. Concentration of enzymes and substrates also influence enzyme activity. Increasing enzyme and substrate concentration will speed up the reaction to a certain point until all the substrates and enzymes are bound together.
Contrary to the lock and key theory, the new theory, the induced fit model, proposed by Daniel Koshland in 1958, is proposed in the following terms. First, for an enzyme action, an accurate orientation is needed for the catalytic group. Second, during binding, the substrate causes a change in the shape of the amino acid at the active site. Lastly, the shape change in enzyme will bring the catalytic group into the proper position, whereas the non-substrate will not. Simply put, the new theory is that when the active site comes into contact with the substrate, the enzyme will attempt to change the shape around the substrate to form a complex, like the action of us catching a tennis ball. The induced fit model also includes prediction on enzyme activators. In general, enzyme activators are chemical compounds that increase the velocity of enzymic reaction by making a deficient molecule act as a substrate by altering the shape of the enzyme.
For example, in the case of a molecule that by itself is too small to induce the proper alignment of catalytic groups, in this case, A and B. A second molecule can bind to the deficient molecule, creating a stable shape with the correct alignment of catalytic groups. So, what disproves lock-and-key theory? Koshland argued that by adding a ligand, which is a substance that forms a complex with a biomolecule, an induced fit shape change could bury some groups and expose others. Koshland uses the phosphoglucomatase and the reactivity of its SH group as a test. Before binding, the enzyme buries the SH group as a ligand. The substrate binds to the enzyme, and a change in shape of enzyme occurs, leading to the exposure of the SH group. Thus, the result is incompatible with the key lock theory, which proves the enzyme's flexibility. Today, almost every enzyme has been shown to undergo significant ligand-induced changes. The finding of extensive confrontational changes in many enzymes is logical, since most enzymes exist in a cytoplasm with many pathways that contains many smaller substrate analogs. For example, trosis, which must be prevented from reacting at the site of larger analogous substrate. Another example is the hexosis. If the specificity failed to exclude smaller analogs, poor yields and bad site direction would occur.
Now, just like us, enzymes do have some weaknesses. Certain chemicals selectively inhibit the action of specific enzymes. Some inhibitors are similar in shape with the normal substrate molecule and compete for admission into the active site. Well, having trouble imagining it? I'm going to show you a scenario. Hey, that's my enzyme! Whatever, don't see your name on it. The green buddy is called competitive inhibitor. It reduces the productivity of enzymes by blocking substrates from entering the active sites. But non-competitive inhibitors don't directly compete with the substrate to bind to the enzyme at the active site. Instead, they halt enzymatic reactions by binding to another part of the enzyme so that the enzyme molecule will change its shape. There are other mechanisms to regulate enzymatic activity too. One mechanism, called cooperativity, amplifies the response of enzymes to substrates. In this mechanism, one substrate molecule prompts an enzyme to act on additional substrate molecules more readily. In this picture on the screen, the binding of one substrate makes the molecule stabilize to become active and capable of binding more substrates.
And the last way to regulate enzymatic activity is feedback inhibition. In feedback inhibition, an end product binds to an enzyme that acts early in a metabolic pathway so that a pathway is halted. See the picture on the screen? Some cells use this 5-step pathway to synthesize the amino acid isoleucine from threonine. As isoleucine accumulates in the cell, it slows down its own synthesis by inhibiting the enzyme for the first step of the pathway. Therefore, feedback inhibition prevents the cell from making more isoleucine than is necessary, and thus wasting our precious chemical resources.